Good evening, everyone. This is your eighth week session with me. In today's session, we'll be looking into the applications of genome editing in treating the human diseases, and in which we'll look into certain disease in which the cell engineering has been incorporated. So, the disease which we will see today are the thalassemia, severe combined immunodeficiency, which is SCID, and hemophilia. Then we'll practice few MCQs. We'll look into the current literature and then I'll take your doubts. So firstly, we'll talk about the key concepts in these topics. So as we all know that with the help of genome editing, we can make precise modifications of the DNA at specific locations. So till now, we have looked into the zinc finger nucleases, TELENs, CRISPR-Cas9, and we have seen that how we can use them for genome engineering, in which we make double-strand breaks, and then depending on the pathway, how the DNA gets repaired, different outputs can be achieved, such as gene could be disrupted, gene could be corrected with a correct donor DNA, or we could also make certain additions of mutations or certain changes we can make at the level of gene. So in that way, we can correct defective genes which are responsible for inherited disease. So with the help of genome editing, we can treat certain disease which otherwise get inherited. So we can correct those genes. So two case studies are of severe combined immunodeficiency, which is KIT, and another one is hemophilia, A, B, and C. And in the later slides, we'll look into the detail of these disease, that what actually happens, which genes get affected, and how it affects the person. Common genome editing tools which are used are CRISPR-Cas9, TELENs, and zinc finger nucleases. So our goal is to permanent and design certain curative therapies for the patients so that we can cure these diseases. So this is kid. This is a normal person, and his immune system is better. He has T cells and B cells which are completely functional. So generally, T cell and B cells are responsible for fighting against infections. So they can fight against infections. But if you are suffering from SCID, which is severe combined immunodeficiency, so it's clear from the name immunodeficiency. So as these cells are responsible for the immunogenic response or immune response, so there is deficiency of these cells, due to which your immune system is compromised. You cannot fight against infections. So you ultimately die if you are not able to get proper medications and proper treatment. So you won't be able to fight against infections. So ultimately it will lead to cell death. Then if we talk about hemophilia, so generally if we have a normal blood vessel, then whenever the damage happens to normal blood vessels, then the bleeding takes place, but soon it gets coagulated. Soon there is clotting of blood. But in case of hemophilia, due to the absence of certain clotting factors, the bleeding gets excessive. It is uncontrollable, and due to which the clotting factors are missing, so it could not get clotted, due to which there is a huge blood loss. So in this way, hemophilia can also lead to the death of a person if it's not cured, if there is excessive bleeding. So let's start with the human immune system. So we'll look into the basic of human immune system. So there are basic cells such as T cells, which mature in thymus, and they are essential for cell-mediated immunity. They activate other immune cells via cytokines. So cytokines are certain chemicals which are released by these T cells, and other cells have receptors to that could accept these cytokines, and they can react accordingly depending on the cytokine response. Then comes B cells, which mature in the bone marrow. So based on the place where they mature, they have got these names. So T cell has got the name T cell because they mature in thymus, whereas B cells, because they mature in bone marrow, so they have got the name B cells. They are responsible for antibody production. So I hope you are aware of the term antigen and antibody. So whenever some foreign particles enter into our body, then our body has an immune system which recognizes whether this particle belongs to self or it is non-self, or is it foreign. So that foreign particle with which our body, which detects our, which is detected by our body, is referred to as antigen. And specific to that antigen, there is an antibody which will bind to that antigen. So that is known as antibody. So in that way, B cells are responsible for production of those antibodies, and they can directly recognize these antigens. So in that way, they will kill those antigens and protect our body from the foreign infectious agents. Then comes natural killer cells, which are also part of our immune system. So they are part of innate immunity. So innate immunity is the one which is by birth, which we get by birth, whereas there is acquired immunity. So whenever we encounter some infection and then our body develops immunity, but later on, whenever that infection again attacks us, so there is memory which is stored in these memory B cells. So that is known as means that is acquired immunity. Means after you're exposed to certain infection, then the immunity which you have gained. Whereas innate is by birth. And then another example could be, for example, we are getting vaccines. So that is acquired immunity, which is not generated by our body, that are acquired from outside. So natural killer cells are part of innate immunity. They destroy virus infected and cancerous cells, release cytotoxic molecules like perforins and granzymes. So this is the way of our body fighting against infection. So they release certain chemicals. As we have seen in case of T cells, they were cytokines. Similarly, in case of NK cells, the chemicals which are released, which are cytotoxic, they are perforins and granzymes. So we all know that how the enzymes work. So these granzymes and perforins, these mainly make the pores in the membrane of the cells which are affected by cancer or virus. So generally our aim is to kill those cells to control the spread of the infection. So generally this is the mechanism that how with the help of release of certain chemical compounds, natural killer cells fight against those cancerous or infected cells. And SCID involves dysfunction or absence of these cells. So in SCID, which is the severe combined immunodeficiency, in which there is a severe effect on the immune cells have been observed. So these all main immune cells which are responsible to fight against infection, all these cells get compromised. They are either absent or they do not function properly in case of SCID. So here I have given in detail that what are different cells in the body, and these are the lymphoid organs which are responsible for these immune cells. So and these are the antibodies. So there are generally 5 types of antibodies which are present in our body. Certain antibodies are just smaller in size, whereas another antibodies which exist in different units get combined. So multiple units. So multimeric, which is IgM, and then IgA, IgG, IgD, IgE, IgG. So depending on what kind of chemical or antigen is present, so respectively these antibodies will fight against them. And these are the T cells, B cells, and NK cells released from these LPCs, which are the lymphocyte progenitor cells, whereas this is hematopoietic stem cell, which is responsible for the blood cells, such as eosinophils, basophils, monocytes, neutrophils. So these monocytes further give rise to dendritic cells, macrophages. So these all cells belong to the immune system. They're responsible in the immune system. Now we'll talk about SCID. So it is severe combined immunodeficiency. And as the name suggests, it is a deficiency. It's not like a disease, but it's a deficiency. This is a rare and life-threatening group of genetic disorders, which is characterized by profound defects in T cell development. So, whenever somebody is affected with SCID, so he will always be having the issues with the T cell development. So, they are always affected. Whereas B cells and natural killer cells functions may also be affected. So generally, it is a group of genetic disorders. That means there is a range in this. So if we talk about other diseases, then we know exactly which gene is responsible. But in the case of SCID, as it is a deficiency or is it a disorder, so in that way, multiple genes are responsible for this disease. So major defect which is observed in the case of the patient suffering from SCID is the T cell development. So in all of them, T cells are always compromised, they are not developed properly. Whereas if we talk about B cell and natural killer cells, then it can vary from person to person. In some people, they are just affected, their function is affected, they are not completely affected, but some of the functionality is affected. Whereas in other patients, they could be completely non-functional. So it depends. So infants with SCID appear normal at birth, but develop life-threatening infections within months. And without intervention, if no cure is being developed, so death can occur usually within first 2 years of the life. So this is to show you that how the hematopoietic stem cells, which are obtained from the bone marrow, so they can give rise to pre-B cells and pre-T cells. So pre means which are the precursor cells, which will later on develop into mature B cells and mature T cells. So in the case of SCID, these B cells if this is particular disorder in which the B cells do not get properly matured into the complete B cells. So this will lead to certain bone defects. Whereas similarly in the case of pre-T cells, if SCID happens, then pre-T cells will not be able to get converted into the mature T cells. There will be issue with the thymus because thymus is an organ in which T cells get matured. So we have seen earlier. So now due to this SCID, the T cells will not be able to get matured. And there is another syndrome which is Dye-George syndrome. So in that case, the T cells could not get matured properly, which are responsible for the cell-mediated immunity. So if your T cells are compromised or if they are not properly developed, so there will be no cell-mediated immunity. That means whenever viral or fungal or protozoan infections will take place, so your body will not be able to fight against them. So you will eventually die. So that's what was observed in the case of this infant who was suffering from SCID. So initially he was fine, but soon in within 2 years he died. So because of this lack of immunity, lack of T cells. Now we'll talk about that what are the genetic bases of this SCID. So SCID is caused by mutations in a single gene. So it is a monogenic disease and there are different inheritance patterns. So whenever there is mutations in a single gene, that's why it is referred to as monogenic, but that doesn't mean that only this single gene is responsible for SCID because it is a disorder. So it could be inherited in the form of autosomal recessive. Both the copies of gene are defective. So in this case, the gene is present on the autosomes. Whereas if we talk about X-linked inheritance, so the mutation carried on the X chromosome. So there is another gene which is present on X chromosome. If there is defect in that particular gene, then its inheritance is X-linked. So it can have both these inheritance patterns depending on that where the gene is present, which get affected. So multiple genes can give rise to this particular disorder. So most common genes which get affected during SCID are IL2RG, which is common gamma chain, most common X-linked SCID. About 50% of cases in which the person is suffering from SCID, they have generally the loss of this particular gamma chain and this IL2RG gene. And this, as this gene is present in the X chromosome, so this type of inheritance, so whenever this disease will get inherited into the means kids, so it will be X-linked inheritance pattern. Whereas then comes ADA, which is adenosine deaminase, and about 15% of SCID cases belong to this gene defects. Then RAG1 and RAG2, which is responsible for VDJ recombination. So these defects in this VDJ recombination are generally um, means give rise to SCID. So this inheritance pattern will be autosomal recessive. Similarly, JAK3, IL7R alpha, and FOXN. So these gene defects can also give rise to SCID, but the defects are less common. Okay, so this shows the inheritance pattern. So as if it is X-linked recessive, that means whenever a disease is X-linked, that means it is that gene is present on the X chromosome. Recessive means whenever this particular X chromosome, whenever it is in its recessive form, then only the disease, then only the individual will get affected. Otherwise, it will not get affected. So let's talk about the unaffected father and the carrier mother. So because female have two X chromosomes, so female will only get affected when both the X chromosomes have that defective gene. If only one chromosome is affected, then in that case, the female will be a carrier. And as we all know that the males have only one single copy of X chromosome. So if male get that affected X chromosome in which the gene is defective, so male will always get the disease. So now you can see in this case, this is the unaffected copy in which there is the light colored box you can see, and whereas if it is affected, then it is blue color box. So now this son has got the X chromosome from his father, this copy of X chromosome from his father. So he will not get affected because this is a unaffected copy. Whereas this daughter has received one X chromosome from the mother and another X chromosome. Sorry. So in this case, as I'm so sorry, I mentioned. So this X chromosome was obtained from the mother, which was the unaffected copy, and Y chromosome was obtained from the father in case of unaffected son. Whereas in case of carrier daughter, the unaffected X chromosome copy is obtained from father, but from mother, the affected X chromosome is present. So in this case, because only one X chromosome has the defective gene, So as it is X-linked recessive, so female will only be a carrier and she will not be affected. Whereas in this case, the X chromosome, which is having the defected gene, is obtained from the mother and Y from the father. So in this case, the affected copy is present. And as in son, there is only single X chromosome, so he will always get affected if the single X chromosome has a defected gene. And similar goes for this unaffected daughter. So in this case, both the X chromosomes are unaffected. So that's why she is unaffected. Similar goes here. So in this case, father is affected. So in this case, in all the female, because father gives the X chromosome, so in all the female will always be the carrier because mother is unaffected. So females will be carriers, whereas males will be always unaffected because their mother has both the corrected copy and son will only receive X chromosome from their mother and not from father. So in this case, all the children are unaffected from the disease, although the females are the carrier in this case because they have received X chromosome affected gene from the father. So I hope it will be clear now. X-linked recessive. Similar goes for the autosomal recessive, but in this case, because in the case of X-linked, there was the biasness between son and daughter, but in case of autosomal recessive, there will be no biasness. It will depend that how many copies of the recessive gene are inherited. So if both the autosomal pair has the defective gene, then only the disease phenotype will be observed. Whereas if only one copy is defective, then there will be no disease. And in this case, the carrier concept will go away. Okay, just give me a moment. Okay, so let's continue. Now we will talk about the symptoms of SCID. So early signs of SCID are start appearing after 2 to 3 months once the maternal antibodies decline. Now what happens is that whenever a child is born, then we inherit some antibodies from our mother. And that kind of immunity is referred to as innate immunity. So after 2 3 months, that immunity, that antibodies start to decline because that antibodies are continuously fighting against infections. Because in the case of SCID, our T cells are compromised. So we do not have cell-mediated immunity with us. We only have the immunity which we have inherited from our mother. So after 2 3 months, it will decline. So the symptoms will be recurrent severe infections. So you will see that the child will severely suffer from certain infections such as pneumonia, sepsis, then there will be chronic diarrhea and poor nutrient absorption, oral thrush, that means persistent fungal infections, skin rashes and eczema like lesions, failure to thrive, there will be poor weight gain and growth. And if there is no treatment done, then most of the infant will not survive after 1 to 2 years, so 12 to 24 months. So these are some of the common symptoms. Now we can classify SCID by the phenotype. So phenotype means what what we observe. So SCID type is T negative, B negative. That means T cells are absent, B cells are absent. Natural killer cells could either be normal or they could be low. Example mutations. So the genes such as RAG1, RAG2, ADA, AK2, if there is mutation in these genes, so all these genes are responsible for the T and B cell development. So if there is mutation in these genes, T cell and B cells will be absent. That's why this type of SCID is T negative, B negative. Then comes T negative, B positive, NK negative. So keep this thing in mind that this minus symbol means that what are the cells which are not present, plus means what are the cells which are present. So in this case, because it is T negative and NK negative, that means both the T cells and natural killer cells will be absent. B positive means B cells will be present. So this happens if the mutation is in IL2RG and JAK3 genes. Then comes T minus, B positive, NK positive. That means only T cells are absent, but B cells are present and NK cells are also present. This happens if there is defect in IL7R alpha gene. Then there are some other forms in which T cells could be variable, B cells will be variable, NKs will be variable. And the gene mutations, the mutations are observed in the genes such as FOXN1 DiGeorge syndrome. So as you can see that in all these cases, T cells are always affected. So keep this thing in mind that SCID is a disorder. And in this disorder, T cells are always compromised. T cells are always absent. Whereas in these genes, it could be variable. That means they could be non-functional, not completely absent. Whereas B cells and NK cells, they can vary. They could either be absent or present, but T cells are always compromised. So in the case of SCID, there will always be the poor immune system because the main immune cell, which is T cell, is missing. And similar thing is shown here, that which of the genes give rise to what kind of uh, phenotype of SCID, and what genes are responsible for this conversion. So if there is loss of these genes or defects in these genes, then this conversion will not take place, which will ultimately lead to the absence of those cells. And similarly, this is also what I have mentioned previously. So if you're interested, you can go through this in detail. And I have also mentioned the paper from where I have obtained this figure, so you can read that. Now we will look into the major mechanisms which leads to this skid. So first one is hematopoietic precursor survival defects. So hematopoietic precursor means hematopoietic. So hematopoiesis is the process of synthesis of the blood cells. So hematopoietic precursor means those cells which give rise to blood cells. So if there is defect in those precursors, so such as AK2 mutation which causes reticular dysgenesis. So this could also lead to SCID. Then defects in VDJ recombination, such as RAG1 and RAG2 mutations. So these can also lead to SCID. Then comes cytokine signaling pathway defects. So as we all know that the T cells are responsible to secrete cytokines. So if there is issues in the signaling pathway, then also SCID will be observed. And the mutations are observed in IL2RG, JAK3 and IL7R alpha mutations. Toxic metabolite accumulation. So if there is accumulation of ADA or PNP deficiency can lead to the SCID. Thymic developmental abnormalities. So if thymus is not properly developed, then also T cells cannot get properly matured. So FOXN1 mutations and Digeorge syndrome are responsible for the thymic improper development of thymus, due to which the T cells do not get matured and hence it leads to SCID. Yeah, so this is what is mentioned here. Now, hematopoietic precursor survival defects. So, AK2 gene mutation leads to reticular dysgenesis, which is a rare form of SCID, failure of early myeloid differentiation, and both innate and adaptive immune cells are absent. So, innate, as I mentioned, so which are from birth, whereas adaptive means which immune cells which are gained after the infection has happened. So, generally, B cells are the innate cells, whereas the memory cells, memory B cells, are the adaptive immune cells, which generated once the B cells encounter infection. So both of them are absent. And clinical features are aleukocytosis. That means the WBC count will be very low, less than 400 per microliters. And clinical features are that it leads to severe and recurrent infections immediately after the birth. So these features will tell you that the child must be suffering from SCID and there must be some mutation in this AK2 gene. And it is usually fatal within days to weeks without intervention. So if treatment is not done, then it could be fatal. It could lead to the death. So this is AK2 deficiency. So this person has this deficiency of this gene. So it will lead to recurrent sinopulmonary infections, and which is also known as hypergamma globulinemia. So two new homozygous AK2 variants exist. One is AK2G100S and another one is AK2A182D, which give rise to normal T cells and defective B cells. So now T cells are all right, but B cells are defective and there is full dependency on mitochondria. So no ATP for cell activation, reduced proliferation and survival. So in that way, B cells will be very less or they are get absent, due to which it leads to reticular dysgenesis. And this AK2 is the adenylate kinase 2. So if you want to read it in detail, then you can go through this paper in which they have mentioned that how the mutation in this gene leads to reticular dysgenesis, which is also known as aleukocytosis, which means low count of WBCs. And then there is another paper in which they have looked into the AK2 deficient mice, recapitulates the impaired lymphopoiesis of reticular dysgenesis patients, but also lack erythropoiesis. So as we have mentioned that this AK2 gene It is leading to the, because mutation in this gene or loss of this gene leads to low WBC count. But in mice, scientists have also seen that not just WBC but RBC count was also less. So this is the wild type mice, human versus mouse. And then they have compared it with AK2 knockout, in which they have knocked out this gene, they have removed this gene so that this could not give rise to the protein. So in the case of human, if it is wild type, then normally erythroid progenitor cell give rise to mature erythrocyte, that means mature RBCs. Similarly, in case of mouse, when this gene is present, it give rise to mature RBCs. Whereas if it was knockout, then in case of humans, the erythropoid progenitor cells were giving rise to mature RBCs. They were not affected. Whereas in case of mouse, the erythroid progenitor cells could not give rise to mature erythrocytes. So that means in case of mice, this AK2 deficient mice, not just they loses WBCs, but they also do not produces RBCs without the AK2 gene. Now the second reason which leads to SCID is VDG recombination. So if there is defect in this VDG recombination, so VDJ stands for variability, diversity, joining, which give rise to variable isoform of these antibodies. So it generates diversity in antigen receptors, which are TCRs and BCRs, which are present on the surface of antibodies. So the process is that random selection of variable, diversity, and joining gene segments. So these are the gene segments, which is VVV, which are variable gene segment. This is DD, and then this is JJ. So they could get rearranged, and this will give rise to the variability in the receptors, antigenic receptors. So DNA breaks and rejoined by the specialized enzymes. Enzymes which are involved in this VDJ rearrangement are the RAG1 and RAG2, which initiate the DNA cleavage, and then NHEJ repair proteins such as Artemis, DNA PKCs, and XRCC4 and ligase 4. So if there is failure, then no functional receptors and apoptosis of T cells and B cells will take place. Yeah, so similar thing is mentioned here, that how after synapses these can recombine. So there is recombination which is taking place after the cleavage, they get rearranged. So now this segment got removed with the help of RAG1 and RAG2. There is cleavage of this segment, this is removed, and then there will be end joining with the help of these enzymes. So what happened, that the DDJ, so this is the means the intermediate, the middle fragments get lost. So this particular fragment, which was DJ, it is lost. This one region is lost, and then this D is now combined to the half of this D and the other J from here. So in this way, this is known as VDJ recombination, in which the VDJ units are recombining in different combinations, due to which they generate diversity in the antigenic receptors. So if there is diverse antigen receptors, so they can bind to different antigens which are present in our body, or whenever some foreign particle attacks us. But if there is problem in this VDJ recombination, that means we will not have enough diversity of antigenic receptors. So our antibodies will not be able to fight against all those antigens. So which will ultimately leads to the apoptosis of T and B cells. Then comes the RAG mutations and Omenn syndrome. So RAG mutation means the gene RAG1 and RAG2, there is mutation in those genes, and that leads to T negative, B negative SCID. That means both the T cells and B cells are absent. Most mutations occur in the catalytic core domains. So in these RAG, if we talk about where the mutation happens, so in the catalytic core domains, for example, which is shown here, that these domains are responsible for the catalysis. Then comes Omenn syndrome. So it is a partial defect, which is also known as hypomorphic mutations. It leads to oligoclonal activated T cells, autoimmune symptoms such as erythroderma, in which there is a skin rash, hepatosplenomegaly, and chronic diarrhea, eosinophila, and elevated IgE. So there is elevated IgE antibody, which is known as, so this all happens in the Omenn syndrome. So similarly, here it's shown that how the RAG mutations can give rise to SCID also and Omenn syndrome also. So if there is abnormal RAG function, so if there is mutation in RAG1 and RAG2 gene, then that means your RAG protein will be non-functional. It will not function properly, so which will lead to SCID. And we have seen that how this particular protein is responsible for the maturation of T and B cells. So they will not get matured, hence SCID will take place. Similarly, if there is mutation, then the RAG functioning will be abnormal and there will be GVHD-like phenotype, eosinophilia with elevated IgE levels, and it will give rise to Omenn syndrome. Now we will talk about cytokine signaling defects. So we are one by one looking into different things which leads to SCID. The third one is cytokine signaling defects. Cytokine receptors are essential for immune cell growth and differentiation. So common mutations are IL-2RG. If there is mutation in this gene, common gamma chain, most common cause of SCID. Then comes JAK3, which is responsible for the intracellular signaling molecule, and IL-7R alpha, which is important for the T cell development. So it results in the T cells and natural killer cell development impaired, and B cells may be present, but they are non-functional in the case of cytokine signaling defect. So if there is normal cytokine signaling, then the cytokines can bind to the cytokine receptors, and there will be normal number of T cells, B cells, and natural killer cells. Whereas if there is gamma chain SCID, that means in this case IL-2RG. Okay, so in that case, there is decreased number of natural killer cells. Similarly, in the case of IL-7, similarly, this particular, there is also decreased T cell number. So if there is defect in this particular gene, so as you can see, the alpha, beta, and gamma C chains must be present. So if there is loss of any one chain, such as in this case, gamma C chain is not present. In this case also, gamma C chain is defected. So in both of these cases, when there is defects in the gamma chain, so there are decrease of the, in this case, natural killer cells are decreased in IL-15 and IL-7. The T cell numbers get decreased. And then this comes JAK3 SCID. So where intramolecular signaling, this is the intramolecular signaling molecule, which is JAK3. So if JAK3 is missing, then it will lead to decreased natural killer cell numbers in case of IL-15. In case of IL-7, if JAK3 is missing, it leads to decrease in the T cell numbers. And similar in this case of IL-7, so IL-7, 7R SCID. So in this case, alpha chain is missing, which leads to decreased T cell numbers. So this outcome is mentioned, and the thing which is mentioned, which is shown in the without color, so that thing, that means there is defect in that particular component. Similarly here also, this is previously green, but now it's white. That means this particular JAK3 gene is defected. There is an example of X-linked SCID of the, so the example is of the bubble boy, and this is the most famous SCID case, and the person, his name is David Vetter. He survived, he lived between 1971 to 1984, and he was born with the IL-2RG mutations, due to which there was completely T cells and NK cells were completely absent. So he lived inside a sterile plastic bubble to avoid the infections. So if T cells, natural killer cells, if they are defected, that means you will not be able to fight against infections. So one way is that either you get the immunity from outside, or another thing is to prevent. So this is the prevention. So in this case, the baby was inside the sterile plastic bubble to prevent him from all the infections, so that he do not require these immunogenic cells, these immune cells. So highlighted the need for gene therapy and public awareness of SCID. So from here, it came into the mind that we need certain treatments so that we can cure these genes, which such as IL-2RG gene, so that we can cure this mutation and we can treat this kid. So sadly, in 1984, 4 months after receiving a bone marrow transfusion, David died from the lymphoma, which is a cancer later determined to have been introduced into his system by Epstein-Barr virus. So this virus is responsible for the lymphoma, which is the cancer of lymphatic system. So Carol Ann, who is the mother of this David Vetter, so Through her work with the IDFs as a long-time member of Foundation Boards of Trustees, Carol Ann mentioned, and she believed that because of what was learned from David Gellen's life and death, many children with SCID have since been diagnosed early, received bone marrow transplants, and now lead the healthy lives. So, in her words, David was a great blessing to our family and to the world. So, she believed that after the case of David Vetter, previously it was not known that how to cure it. But later on, it was realized that with the bone marrow transplant, we can give those cells and we can give progenitor cells because we know that the stem cells are the cells which can almost give rise to any cell in our body. So if we give the bone marrow cells, bone marrow transplant, if bone marrow transplant is done, so the precursor cells can give rise to the lymphoid progenitor cells and which will further give rise to proper functional T and NK cells. So from this case, many children got diagnosed early and treatment was developed so that they survived. So next reason is toxic metabolite accumulation, such as adenosine deaminase deficiency. So toxic accumulation of DATP and adenosine, which leads to the apoptosis of developing lymphocytes, and it accounts for 15% of SCID cases. So in the absence of ADA, the DATP and adenosine accumulates in toxic concentrations. So as you can see here, that there is increase in their number. So they interfere with the DNA maintenance, which are responsible for lymphocyte development. So if there is imbalance, so lymphocytes will not be properly developed and it will lead to frequent infections and immune dysregulation. Then comes PNP deficiency. So excess deoxyguanosine toxic to T cells specifically. So if PNP will not be there, so which will give rise to lot and lot of deoxyguanosine, which will get toxic. So these are treatable with the enzyme replacement therapy or gene therapy. So if we supply these enzymes, then we can treat it. So adenosine deaminase is encoded via the gene on chromosome number 20. And if there is mutation in this gene, this ADA gene, then it uh, give rise to the accumulation of DATP to toxic concentration in our cell. So this inheritance pattern is autosomal recessive, and in this case, 25% is the chance that the children will be completely healthy, 50% is the chance that children will be carrier, and 25% the chance that he will be affected with the ADA skip. Okay, now we'll talk about the thymic developmental defects. So in this case, if the thymus will not be properly developed, then also skid can arise. So one example is of Dijot syndrome, in which there is malformation of thymus and parathyroid glands due to some developmental defects. So due to defects in the thymus, T cells will not be able to mature, so there will be either partial absence or complete absence of T cell development, which will give rise to skip. Now comes the FOXN1 mutations. So these also leads to Newt/Skid syndrome. In this case, there is the absence of thymic epithelium, so T cells will not get matured. So this is the another pictorial representation which shows that how the Dijot syndrome, which is also known as thymic aplasia, leads to T cell deficiency. So microorganisms will not leave the T cells, and T cell thinks that I wish thymus was alive so that I could get matured, but this is not mature. So this will lead to the recurrent infections. And this is the thymic gland which generates mature T cells. So it says that I miss protection of T cells because now it's not able to mature them. Okay, then there will be some facial abnormalities such as cleft lip and cardiac abnormalities, absence of parathyroid gland, so low calcium, which can ultimately lead to tetany, and then 22 microdeletion detected by FISH, absent 3 and 4 pharyngeal pouch, absent thymus and parathyroid glands, no thymic shadow on the chest X-ray. So yeah, so this is the one of the representation, and another one is of the FOXN1 mutations. So the patient genotypes and phenotypes. So in the case of normal FOXN1 gene, so it was observed on the mouse models. So this is the normal thymus and this is the normal fur. But in case when the FOXN1 compound hit the mutations, so in this case it gives rise to skid. Now if we take samples of thymus and fur section, then in this case, this is the thymus which is quite short. This is referred to as hypothymous. And this is the fur which is darker. Then comes FOXN1 AR mutations. So this give rise to nude or nude slash skid. So in this case, again there is the hypothymous, but the fur is nude. That's why this is referred to as nude skid. Okay, so now we are done with the skid. Now we'll look into the hemophilia. So hemophilia is a genetic bleeding disorder which is caused by the clotting factor deficiency, and it results in the inability to form the stable blood clots after injury. There are different types of hemophilia: hemophilia A, hemophilia B, and hemophilia C. So it depends that which of the clotting factor is deficient. Depending on that, the different names of hemophilia are given. So in the case of hemophilia A, it happens when there is deficiency of factor 18. Whereas in the case of hemophilia B, it happens if there is deficiency of, sorry, not 18, factor F8. And in this case, if the deficiency in the factor 9. And in the case of hemophilia C, the factor 11 deficiency, which is rare and autosomal recessive. So 80% of the cases happens if there is deficiency of factor 8, which is the clotting factor 8. Okay, now these are the different blood clotting factors which are responsible to clot the site where the bleeding has happened, where there is wound. So platelet move rapidly on the site of the lesion. Lesion means wherever there is a wound or uh, some injury. So cascade reaction of coagulation factors take place. Cascade means series of reactions, and this is the lesion. Now there will be formation of fibrin, which will going to settle on the platelet thrombus, stabilizing the consistency. And this is the red thrombus formation that permanently stops the bleeding. And then action of plasmin that models the thrombus and dissolve the excessive proliferation. And then fibroblast synthesizes a new tissue, endothelial, restoring the integrity of the vessel wall. So this is how the blood clotting takes place. Now, in the case of hemophilia, if the normal blood cell is there, if there is some tear, then platelets and clotting factors will come and they will clot the blood here, and ultimately fibroblasts will generate a new layer of skin. Whereas in case of hemophilia, due to missing of some of the clotting factors, this cascade events will not take place and it will lead to the uncontrolled bleeding. And if nothing is done, then person can die. Now we will look into the genetics of hemophilia. So hemophilia A and B are the X-linked recessive disorders. So because they are X-linked recessive, so they primarily affect males, because whenever their mother is affected or mother is carrier, then the males will always get affected because they will inherit X chromosome from their mother. So males have one X chromosome, so mutation shows full effect. Females are carriers with one normal and one defective X. So the example is of Queen Victoria. She was a carrier of hemophilia B. So as I talked, that hemophilia B happens due to the deficiency of factor 9. Okay, similar pattern has been shown here. So we have already talked about extreme recessive. So similar thing is observed here, and if you're interested, you can go through this. Yeah, so this is the history of hemophilia. So it was first described in 1803 by John Conrad Otto, and this is called a royal disease because it was first observed in the royal families. So European royal families, it spread through them. That's why it was referred to as the royal disease. And Queen Victoria's descendants carried the defective gene across multiple royal bloodlines. So this is their family tree. So the photos which are having the border of blue color, they are suffering from hemophilia, whereas the pink borders represents the carriers of hemophilia. So it began with Queen Victoria. She was the carrier of hemophilia B, and then she has passed on this defective gene on X chromosome to her two daughters, Princess Alice and Princess Beatrice, whereas the Prince Leopold, So he got the defected X chromosome from his mother, so he was affected with hemophilia. And later on, this same defective gene was passed on to the later progenies, and similarly, Prince Frederick, Prince Leopold, and Prince Maurice, they all get affected. So mostly, as you can see, the prince were the ones who got affected, whereas the females were the ones who were the carriers. And then their later generations all were affected with hemophilia because all were the sons. So from here we can see that the X-linked disorders, they are quite biased towards sons because they only have single X chromosome. So many cases if that particular X chromosome is defective, then they will always be the ones who will get affected from the disease. So if we talk about the global prevalence, then hemophilia A will be observed in 1 in 5,000 male births worldwide. Hemophilia V, 1 in 30,000 male births. Hemophilia C is rare and more common in specific populations like Ashkenazi Jews. So majorly the hemophilia which is observed is the hemophilia A followed by hemophilia B. Yeah, so this is the report of the World Federation of Hemophilia. So they have mentioned the statistics. So if you're interested, you can go through this. This was published in September 2019. Now we can classify the severity of this hemophilia. So severity is mild, moderate, and severe. So depending on the levels of factor 8 and factor 9, the severity can vary. So if it is 5 to 50 international unit per DL, so then it is bleeding after the trauma or surgery. This is the symptoms. Moderate means 1 to 5 international unit per DL. This leads to the symptoms are occasional spontaneous bleeding, and severe when there is less than 1 international unit per DL of these factors, so frequent spontaneous joint and muscle bleeds. So if the concentration of factor 8 and factor 9 are very low, then that case is the severe hemophilic condition. So based on the levels of factor 8 protein in the blood, the severity can vary. So type A or classic hemophilia, 80 to 85% of the hemophilia cases in which the factor 8 is less. Type B is also known as Christmas disease, in which 15 to 20% of hemophilia cases belongs to type B, in which the level of factor 9 is reduced. Now we'll talk about the coagulation cascade, that how different factors are involved. So there are two pathways of coagulation. One is intrinsic and another one is extrinsic. So intrinsic pathway involves factor 8 and factor 9 and it is triggered by internal vessel injury. Extrinsic pathways are triggered by tissue factors and both converge to activate factor 10 and lead to fibrin float formation. So intrinsic pathways are the collagen, kallikrein and HMWK, which converts Hegman factor, which is factor 7, into factor 7A. Then this factor 7 is responsible to convert factor 11 into factor 11A. And then this factor 11A, along with the calcium ions, it is responsible to convert factor 9 into factor 9A. And this factor 9A is responsible to start this cascade. So now thrombin is required to convert factor 8 into factor 8A. And this factor 9A, 8A, along with the calcium, they are responsible to convert factor 10 into factor 10A. This factor 10A along with factor 5A and calcium, it leads to conversion of factor 2 to factor 2A. And then this 2A is responsible to convert factor 1 to factor 1A, which is fibrin. And this along with the calcium and factor 8A is responsible to generate the stable fibrin clot. And this is the extrinsic pathway in which if there is damage to endothelial tissues, then factor 3 is converted to factor 3A and factor 7 is converted to factor 7A and they also activate factor 10. So this is the extrinsic pathway and this is the intrinsic pathway. Collagen, kallikrein and HMWK, they are responsible to initiate the intrinsic pathway, whereas damage is responsible to initiate extrinsic pathway. So this is the series of reactions and ultimately their all aim is to convert factor 1A and factor 8A to generate a stable fibrin clot. So coagulation disorders can either cause excessive or inadequate clotting. Deficiency in more than one clotting factors leads to hemophilia A, hemophilia B, hemophilia C, von Willebrand disease, and vitamin K deficiency. Now we will look into the role of factor 8, because we have mentioned that out of factor 8 and 9, factor 8 is most important because hemophilia A is caused due to factor 8 deficiencies. So it serves as a cofactor for factor 9A in the intrinsic pathway, stabilized in blood by von Willebrand factor, which is VWF. Activated factor 8 and factor 9A generate teenase complex, which activate factor 10. So when we were looking into the intrinsic pathway, then we have seen that how factor 7, factor 9, factor 11, factor 9, factor 8 activates the factor 10, and then factor 5 and 10 generates thrombin, which is factor 1, along with the fibrinogen generates the fibrin clot. Similarly, in extrinsic pathway, we have seen that how factor 7 got activated and it also activates factor 10. Yeah, similar mechanism is shown here. So this tells you that what is the role of factor 8. Because if factor 8 got missed, then it will not be able to activate factor 10, which is further responsible for this downstream process. Now this factor 8 gene is present on the X chromosome, and the structure is that this gene is composed of total of 26 exons and one large intron, which is intron 22, and the mutations is in the intron 22 inversion, 50% of severe hemophilia A cases. So this particular intron, if it gets inverted, so as you can see here, that this is the region where there is inversion. Okay, so due to this inversion, it leads to severe condition of hemophilia A. Missense mutations cause reduced F8 activity. So these are two ways in which we will be able to see the factor 8 activity being decreased. So one is that due to the inversion of intron 22 or due to the missense mutations, which can also give rise to reduced factor 8 activities. And then factor 9 genes. So the location is on the X chromosome Q arm 27.1. So this tells you the position, that where in the X chromosome, which arm, and at what position. So structure is, it has 8 exons spanning 34 kb, and protein includes signal peptide, GLA domain. So this is what I'm talking about. So signal P, signal peptide, GLA domain, and EGF domains, and serine protease domain. Now mutation types. So hemophilia A, we have seen that if there is inversion of intron 22, or if there is nonsense or missense mutations, or if there are small deletions or insertions, so these all leads to the defects in the factor 8 gene, which give rise to hemophilia A. If we talk about hemophilia B, then if there are point mutations, in 73% of cases we have seen that point mutation leads to hemophilia B, or large deletions, rare but severe. Okay, so we have looked into that how the 8 exons are present and what are the different domains which are present in these genes. So in the factor 9, if there is point mutation or large deletion, so it leads to hemophilia B. This is the another paper which talks about both the hemophilia A and B, and what are the molecular and clinical similarities among them, and what are the differences among them. So if you're interested, you can go into it. It is a detailed paper. Now, how we can use genome editing to treat these disorders. So genome editing for SCID. So one review is the ex vivo gene transfer and correction for cell-based therapies. So this tells you that how with the ex vivo gene therapy we can treat SCID. So patients' hematopoietic stem cells are isolated. CRISPR or other editing tools are used to repair the defective IL-2RG or ADA genes. So depending on the type of SCID, that which genes are affected, so different therapies can be used. So in this case, CRISPR or other editing tools such as ZF and TELENs can be used to repair the defective gene. So if there is issue in the IL-2RG gene, or if there is issues in the ADA gene, so we can cure them. And we have already discussed in previous sessions that how the CRISPR and ZFNs and all those nucleases, how they work. So they will work accordingly. They will repair these defective genes. So we will use homology dependent repair in the end. After the double strand break takes place, we'll provide a donor DNA which contains corrected copy of these genes. So in this way, CRISPR will be able to cure this defect. So corrected cells will be reinfused and the immune function will be restored. So benefit is that it is a permanent cure with patient's own cells and it avoids immune rejection. So this type of method is immune ex vivo gene transfer. So similar thing is shown here that if a child is suffering from skin, we take his HSPs, which are the hematopoietic stem cells. They are isolated and then the gene therapy is done. So after the gene therapy, we will see that what is the defect. So we'll treat that defect. And now, so previously it was defected, so thymus was not completely formed, due to which T cells were not developed. But now, because we have corrected the copy, so after the therapy, now thymus is matured, thymus is well functional. It will mature the T cells, and then after the maturation of T cells, they will give rise to T lymphocytes. Natural killer cells get developed. B lymphocytes will be formed. So in that way, after the therapy, the patients will, this particular cells which are corrected will be infused into the patient and patient can survive. So this is ex vivo gene transfer because after the cells have been isolated and externally they are treated. And in this case, because they are obtained from the same patient, so the chances of immune rejection are less. Now, genome editing for the hemophilia. So, editing targets will be factor 8 and factor 9, depending on that what kind of hemophilia person is suffering from. So, strategies are simple here. We will correct the mutations in situ, insert functional copy into the safe harbor locus. For example, AAV S1 site. So, because we know that when we insert these uh, corrected copies into the cells, then we need certain constructs. So in this case, the adeno-associated virus is generally used. So which is AAV site S1, where we insert the functional copy. So animal models like dogs and mice show long-term clotting factor production because this was checked in them after the single treatment. Okay, so in this case, we enter the complete functional copy of the gene artificially into the body with the help of this viral delivery. So liver specific promoters, low dose, one participant, intermediate dose, one participant, high dose, seven participants. So dose dependent increase in factor 8 activity levels, which will reduce the bleeding episodes. In case of hemophilia A, in case of hemophilia B, the gene which we will provide will be the factor 9. So similar thing will be here also. Okay, so now this is about the how we can treat hemophilia A and B with the help of genome editing. And similarly, these are the cell-based gene therapies. So, and CRISPR-Cas9 is used in them to treat the hepatocytes, which are the liver cells. So, clinical trials in progress. So, ADA state in which there is deficiency of ADA, which is adenosine deaminase. So, FDA approved gene therapy treatment, which is Strimvelis. Then X-linked kit, which is so CRISPR research is ongoing on X-linked kit, and trials are going which are trying to correct the IL-2RG gene. Similarly, hemophilia A and B, so clinical trials with the adeno-associated virus vectors are delivering factor 8 and 9, and early results show sustained clotting factor expression. Yeah, so similar thing is mentioned here that we do transfection of AAV293 cells. In this case, AAV will carry the target gene, which will be the gene for these vectors, and then directly delivery on the patient. So patient will give rise to the protein factor 8 and factor 9 proteins, which will do the proper clothing. Now, viral vector based gene therapy, so in which we use viral vector to transfer the functional copy of the gene into the cells. And in this, it also tells you that how the different milestones are achieved in the case of gene therapy. So, initial exploration phase, rapid development phase, security questioning phase, and nowadays, redevelopment phase. So, if you're interested, you can go through this review. This is a very nice review. So now there are many challenges in the genome editing methods. Strategies to overcome the main challenges of the use of CRISPR-Cas9 as a replacement for the cancer therapy. Okay, now there are various challenges which exist. So first challenge is the off-target effects of CRISPR leading to the unintended mutations. So whenever we are trying to treat something using CRISPR, so we last time we talked about CRISPR-Cas9 system, in which we talked about that it has a single guide RNA which binds to the site which is close to the PAM motif and it cleaves there. But the PAM motif is very short. So what if it binds to some other random site? So which will lead to the off-target effect. That means not to my target site, it is binding to some other site other than my target. Then comes immune reactions against the viral delivery vectors. So the vectors which I am using to deliver this CRISPR-Cas system into the cell, that could our cell could also fight against that viral vector. So there is a chance of immune reaction. Then cost and accessibility of the therapy. So it is quite costly. So not everyone will be able to access that. Ethical concerns, especially with the germline editing. The cases where we are editing the germ cells, so in that case, there are many ethical concerns which arises. Okay, so this also shows the same thing. There's different challenges. So first thing is delivery challenges, that what to use viral vectors, non-viral vectors, because viral vectors we have seen that it generates the immune response. Next challenge is the ethical issues, that whether we should do the changes or we should not do it. So ethical standpoint and morality concept. Then there is chances of the off-targeting. So different bioinformatic tools, CRISPR-Cas9 nicases, and anti-CRISPR proteins. So they will inhibit the off-targeting. We can use them. Then gene polymorphism in cancer. So detecting protein interaction sites, many targets in a single cell, because we know that gene polymorphism means there are multiple gene copies which are present. So in the case of cancer, to detect that where the interaction site exists, which target is the one we should uh, hit, that thing is very difficult. Then autoimmune response against the endogenous Cas9 protein. So in this case, our body also generates some response against this protein. So gene editing in early lifetime and targeting immune privileged organs. So in order to summarize this, SCID and hemophilia, they are classic monogenic diseases with well-characterized mutations. Genome editing offers curative potential. For SCID, restored the immune system function, and for hemophilia, normal floating factor levels. Ongoing research focuses on the safety, precision, and making the therapies accessible worldwide. So, as in this case, this kind of shows you the ex vivo treatment. So, they harvest the hematopoietic stem progenitor cells from mobilized peripheral blood. As soon as the HSP is collected, now they are cultured ex vivo, that means outside the body for the gene transfer or editing. Now they are transferred into the other cells. So this is the conditional recipient. And then after, means externally, when they are treated, when the mutation has been achieved, so endogenous HSPCs from the bone marrow are depleted, which makes space for the ex vivo engineered cells, which are then infused back. So this is known as autologous cell therapy. That means cells are obtained from the same person, and again at the site from where the cells were obtained, there new cells are injected and which are this of the same organisms or same person. So it is autologous cell therapy. And now what kind of things which were modified here? So uh, the genes which are responsible for the maturation of T cells, they are changed. Similarly for the natural kappa cells, natural killer cells, and B cells. So all these genes are repaired. All these genes are, their copies are made right so that they can give rise to these T cells, B cells, and NK cells. Along with this, they can also give rise to erythrocytes, platelets, granulocytes, monocytes, and all of them are responsible for the proper um, functioning of the immune cells, proper functioning of the immune, uh, proper proper showcasing the immune response. Okay, so this is all about today's session. So in this session, we covered about what is SCID. We have looked into the different genes which can give rise to SCID, and based on that, we have also looked into the phenotype of SCID. And then we looked into the hemophilia, and based on which clotting factor is affected, we have seen that which type of hemophilia will be there. And we have also seen that hemophilia A is more prevalent than the other types. And then hemophilia B is the second one, and hemophilia C is quite rare, which is not frequently observed. Then we looked into the different therapies which exist, with which we can cure the SCID as well as hemophilia. And then this is the summary. So let's start with the MCQs. So first MCQ is, which type of thalassemia is characterized by the absence or decreased production of both delta and beta globin chains? Option A, alpha thalassemia. Option B, beta thalassemia. Option C, delta and beta thalassemia. And option D is gamma thalassemia. Okay, so I'll give you some time to think and then we'll talk about the right option. Okay, so now let's see what is the right answer. So the right answer is option C, which is delta beta thalassemia. So it is clear from the question that both the delta and beta globin chains are either absent or decreased. So due to which delta beta thalassemia means it involves reduced or absent production of both delta globin and beta globin chains due to the deletions which affect these genes. Whereas if we talk about alpha thalassemia, then it is caused by the deletion or mutation of just alpha globin genes. And if we talk about beta thalassemia, then it is characterized by partial or complete deficiency in the beta globin chain. So from the name it is clear. So alpha means if there is some problem in the alpha globin gene, beta means if there is deficiency in the beta globin chain, delta beta means if the production of these both the delta and beta globin chains are affected, and gamma means if there is absence or mutation in the gamma globin chain. So from now on, keep this thing in mind. Let's move to the next question. So next question is, what does SCID stand for? Option A, severe complicated immunodeficiency diseases. Option B, severe combined immunoglobulin diseases. Option C, severe combined immunodeficiency diseases. And option D is none of the above. Okay, so this is quite straightforward. So I guess you'll not take much time for this. So right answer is option C, which is severe combined immunodeficiency diseases. So I don't think we need any explanation for this. So let's move to our next question. So next question is, what does HBD stand for? Option A, hematopoietic bleeding disorders. Option B, hereditary bleeding disorders. Option C, hemorrhagic bleeding disorders. And option D is none of the above. Okay, now let's see the right answer. So the right answer is option B, which is hereditary bleeding disorders. So the option one was hematopoietic bleeding disorders. So yeah, I can see that these options are quite confusing because they appear to be similar. So let's look into them one by one. So hematopoietic bleeding disorders, this is incorrect, although HBD is not specifically used to describe bleeding disorders which originate from the hematopoietic cells. So we have seen hematopoietic cells are the ones which give rise to, which are the ones which give rise to the blood cells. Then comes hereditary bleeding disorders. So yes, because these are the bleeding disorders that are inherited genetically. Option C was hemorrhagic bleeding disorders. So this is also incorrect, and this is a descriptive term, but it is not a standard abbreviation. And then none of the above could not be the option. Okay, so the next question is, what is the cause of hereditary persistence of fetal hemoglobin, which is HPFH? Option A, abnormalities in the adult globin genes. Option B, mutations in the fetal globin promoter sequence. Option C is excessive production of adult hemoglobin. And option D is deficiency of fetal hemoglobin during fetal development. Okay, so keep this thing in mind that they have mentioned what causes hereditary persistence of fetal hemoglobin. Okay, so now let's see the right answer. So right answer is option B, which is mutations in the fetal globin promoter sequence. So this HPFH, which is hereditary persistence of fetal hemoglobin, it occurs due to mutations that keeps gamma globin gene active after the birth. So there is mutation in that gene. Now let's look at other options. So first option was abnormalities in the adult globin genes. This is incorrect because it is not caused in the adult globin genes, but by the continued expression of fetal genes. Then third option was excessive production of adult hemoglobin. So this is also incorrect because adult hemoglobin levels are not increased, fetal hemoglobin levels are persisting. So this is also not right. Then last option was deficiency of fetal hemoglobin during the fetal development. So this is also incorrect because here we are saying hereditary persistence of fetal hemoglobin. We're not saying the non-persistence. So that's why all other options are wrong, because in all of them they have mentioned that the fetal hemoglobin is deficient, but it is other way around. It involves persistence, not a deficiency of fetal hemoglobin. Now let's move to our next question. So next question is, which condition is a significant clinical presentation of recombinase activating gene, which is RAG, defects? Option A, Turner syndrome. Option B, Down syndrome. Option C, Omen syndrome. And option D is Morphin syndrome. Okay, so the right answer is option C, which is the Omen syndrome. Now let's look into the other options as well. So the first option was Turner syndrome. So it is incorrect because Turner syndrome is a chromosomal disorder, especially the X chromosome, and it is unrelated to RAG gene defects. Next was Down syndrome. So it is also incorrect because it is caused by the trisomy of chromosome number 21, and it is not related to the mutation in the RAG gene. Whereas Omen syndrome is caused by the hypomorphic mutations in the RAG genes, which leads to SCID, severe immunodeficiency with the autoimmunity. Then if we talk about the Marfan syndrome, then this is also incorrect because it is a connective tissue disorder caused by the fibrillin 1 gene mutations and not the RAG defects. Okay, now let's move to our next question. The next question is, what is the most severe form of severe combined immunodeficiency, that is SCID, that occurs in 15% of SCID patients? So option A is X-linked SCID, option B is JAK3 deficiency SCID, option C is ADA deficiency SCID, and option D is IL-7R alpha deficiency SCID. So you have to tell which one is the most severe form. And this we have already discussed in the previous table when we were talking about different severities of SCID, that when it is, which gene leads to what kind of phenotypes. Okay, let's see the right answer. So the right answer is option C, which is ADA deficiency SCID. So ADA is adenosine deaminase, and its deficiency is the most severe form, occurring in 15% of SCID patients. Now let's look at other options. So first option was X-linked SCID. So it is incorrect because X-linked SCID is the most common form of SCID, but not the severe. JAK3 deficiency SCID. So this is also incorrect because it causes severe immunodeficiency, but it is rare and not the most severe form. If we talk about IL-7R alpha deficiency SCID, then it is also incorrect because it causes SCID due to defective T cell development but it is less severe than ADA deficiency. So from all of them, ADA deficiency SCID is the one which is most severe and it accounts for 15% of the SCID patients. Okay, let's move to next question. So next question is, which genome editing strategy is commonly used to correct the genotype in most genetic blood disease like SCID? Option A, CRISPR-Cas9. Option B, zinc finger nucleases. Option C, TALENs. And option D is homologous recombination. So you need to tell that which of the genome editing strategy is commonly used. And don't get confused by the options. Just keep this thing in mind that they have mentioned genome editing strategy. Okay, now let's see the right answer. So the right answer is option 1, which is CRISPR-Cas9. But I hope you might have got confused with option D. So let's break it down into different options. So let's see. So if we talk about CRISPR-Cas9, then yes, we know that this is the genome editing strategy which is most widely used nowadays for correcting the mutations in genetic blood disorders, including SCID. Now, second option was actually zinc finger nucleases. So this is incorrect because it was previously used and it is now less common due to the low efficiency and high complexity compared to CRISPR. Then comes talens. So similar reason is for talen also. This is also incorrect because it is used in some cases, such as in the case where we want to treat the mitochondrial genes, but it is less popular than CRISPR due to the more complex designs. Because when we're talking about the ZFNs, talens, then we have seen that how to design them. So we have seen their assembly. So we have seen that how we can design the talents and we can decide that to which specific DNA segment it can bind to. Based on that, we need to synthesize them. We need to assemble them. So that is a complex task. So that is a complex designing task. That's why CRISPR-Cas9 is much favored over them. And then last option was homologous recombination. So this is incorrect because it is a natural DNA repair pathway and during genome editing, but not a genome editing tool by itself. So yes, it is used to repair the DNA and to means to replace the faulty gene with the correct copy of the gene. But this is just a process, repair pathway. It is not a tool in itself. That's why this was not right answer. So with this, let's move to next question. So next question is, what type of hemophilia is caused by factor VIII deficiency? So sorry for this mistake. Now option A is hemophilia A, option B is hemophilia B, option C is hemophilia C, and option D is hemophilia 8. So this sometimes when you look into the options, so some options are just to confuse you. So don't get confused. You need to just tell that which hemophilia is caused by factor VIII deficiency. Okay, let's see the right answer. The right answer is option A. So hemophilia A, we have seen that this is caused by the factor VIII, and this is the one which is most common hemophilia, which is majorly observed in about 80% of the cases of hemophilia. It leads to impaired blood clotting. Factor, sorry. So hemophilia B is caused by factor IX deficiency, and this is also known as Christmas disease. Whereas if we talk about hemophilia C, then it is very rare and it is caused by factor XI deficiency, and it is a milder form. And if you talk about hemophilia 8, then it is incorrect. There is no such classification. So this is just to confuse you with the thalassemia, because in thalassemia, when we were talking about different globin gene defects, then we said that alpha thalassemia means if there is defect in the alpha globin gene. So this is just to confuse you. There is no such name which exists. Now let's move to next question. So next question is, which of these is a bispecific monoclonal antibody used for the prevention of bleeding in the patients with HA, that is hemophilia A? Option A, imatinib. Option B, imicizumab. Option C, Serase K. And option D is none of the above. So you need to tell which among them is a bispecific monoclonal antibody, which is used to prevent the bleeding in the case of hemophilia A patients. Okay, so now let's see the right answer. So the right answer is option B, which is amicizumab. It is a bispecific monoclonal antibody which mimics factor VIII activity to prevent the bleeding in hemophilia A. So because we have seen certain papers also when we were talking about today's session, so we have seen that how they have used different factors in the mice and how they have seen that how in those model organisms what is the effect of externally supplying these factors. So similarly, in case of human also, after different clinical trials, it has been observed that if we supply this monoclonal antibody, then it mimics this factor and it can mimic its activity. So it can lead to the treatment of this factor. Now let's look at other options also so that uh, in future, if you find these options, then you must know what they mean. So imatinib is incorrect because it is a tyrosine kinase inhibitor which is used mainly in cancers like CML and not in hemophilia A. So this is the tyrosine kinase inhibitor. Next option was Serase K. This is also not related to hemophilia A treatment. Okay, let's move to next question. So which of these is false regarding thalassemia? Option A, inherited by autosomal recessive manner. Option B, arises due to defects in hemoglobin production. Option C, enlarged spleen is a symptom of thalassemia. And option D, arises due to structural defects in hemoglobin. So you need to tell which one is false. Okay, so now let's see. So the right answer is option D. That means arises due to structural defects in hemoglobin. So this option is false about the thalassemia because thalassemia is caused by the quantitative defects, that means decreased production and not any structural abnormalities. So all other options are right. So first option was inherited by autosomal recessive manner. So yes, it is true because it is inherited in autosomal recessive manner. Option B was arises due to defects in hemoglobin production. So this is also true because the hemoglobin production is reduced due to the absence or reduced production of alpha and beta globin chains. Third option was also right, enlarged spleen. So it is also known as splenomegaly, occurs due to excessive breakdown of abnormal RBCs, which leads to the enlarged spleen. Now let's move to next question. So next question is, what is the main underlying issue in thalassemia? Option A, structural defects in hemoglobin. Option B, decreased or absent production of alpha or beta chains of hemoglobin. Option C, increased production of hemoglobin. Option D is defects in the red blood cell membrane. So you need to tell what is the main underlying issue. So some hint you have already obtained from the previous option, from the previous question only. Okay, now let's see. So the right answer is option B, that is decreased or absent production of alpha or beta chains of hemoglobin. So option A. So we have already seen in the previous question that thalassemia is not due to the any structural defects in hemoglobin and it is not leading to any increased production, but it is vice versa. It is opposite of this. Defects in red blood cell membrane. So this is incorrect because membrane defects are associated with disorders like hereditary spherocytosis and not thalassemia. So now let's move to next question. So next question is, which type of thalassemia involves reduced or completely absent production of both delta and beta globin chains. So this question is again repeating. Option A is alpha thalassemia, option B is beta thalassemia, option C is delta beta thalassemia, and option D is gamma thalassemia. Okay, so now let's see. So the right answer is option C, and we have already discussed about it, so I'll not go into the detail. So next question is, what is the most common genetic cause of alpha thalassemia? Option A, point mutations in the alpha globin gene. Option B, deletion of one or more alpha globin genes. Option C, overexpression of alpha globin genes. And option D is unequal crossing over of beta globin genes. Okay, so let's see. So the right answer is option B. That means deletion of one or more alpha globin genes. So let's see option A. Option A was point mutations in the alpha globin gene. So this is incorrect because point mutations are rare and they're not the primary cause of alpha thalassemia. Second was deletion of one or more alpha globin genes. So this is correct because it is most common cause of alpha thalassemia. And in this deletion of one or more alpha globin gene is being observed, which leads to reduced alpha chain production. Now option C was overexpression of alpha globin genes. So this is also incorrect because alpha thalassemia is due to decreased production and not increased. Last option was unequal crossing over of beta globin genes. So this is also incorrect because this mechanism is associated with beta globin gene rearrangements and not alpha globin gene deletions. So let's move to next question. Next question is, which of the following is a major risk associated with long-term blood transfusions in thalassemia patients? Option A, increased hemoglobin production. Option B, iron overload causing damage to organs like the liver and heart. Option C, decreased chances of infections. Option D, reduced need for iron chelation drugs. So you need to tell what is the major risk with long-term blood transfusions. And this is quite straightforward, I guess. Okay, now let's see what is the right answer. The right answer is option B. So that is iron overload causing the damage to organs like the liver and heart. So option A was increased hemoglobin production. This is correct. Sorry, this is incorrect because transfusions supply extra external hemoglobin, but it do not increase the natural hemoglobin production. Option B was iron overload causing damage to organs. So it is correct because if there is repeated transfusion, it will lead to iron accumulation, which will damage the vital organs. Option C was decreased chances of infection. This is incorrect because blood transfusion can actually increase the risk of infections if not screened properly, but they won't decrease. Option D is reduced need for iron chelation drugs. This is also incorrect because iron chelation therapy becomes necessary to manage the iron overload caused by frequent transfusions. So from here you can take two takeaway messages. First takeaway message is that if there is prolonged transfusion, then it leads to iron overload. And second thing is we need iron chelation drugs to overcome the iron overload. And this long-term blood transfusion can also lead to there are chances of transfection or infection if it's not properly screened, but it do not increase the hemoglobin. So I hope these concepts are now clear. Let's move to next question. So next question is, the most frequent thalassemia mutation found in Southeast Asia is option A, 4 base pair deletion at codon 4142, single point mutation at codon 4142. Option C, 4 base pair insertion at codon 4142. And option D is deletion in the known coding region of HBB gene. Okay, now let's see. So the right answer is option A, that is 4 base pair deletion at codon 4142. This is the most common mutation in Southeast Asia, and it is a 4 base pair deletion at codon, this 4142 of the beta globin gene, which causes a frame shift and beta thalassemia. Option B was single point mutation at codon 4142. It is incorrect because although point mutations occur, but they are less common than 4 base pair deletion. Option C was 4 base pair insertion at codon 4142. So this is also incorrect because mutation is a deletion and not any insertion. Then deletion in known coding regions of HBB genes. So it is also incorrect because this type of deletion does not represent the most frequent mutation in this region. So let's move to next question. Next question is in CRISPR-Cas9 therapy for beta naught thalassemia, what is the function of single-stranded donor DNA? Option A, combine with CRISPR RNA to guide Cas9. Option B, serves as a template to correct the faulty sequence. Option C, creates a double-strand break in the DNA. And option D, activate the PAM site for Cas9 recognition. So you need to tell what is the function of single-stranded donor DNA. Just think for a second and then we'll discuss the right answer. Okay, so let's see. So the right answer is option B, which is serve as a template to correct the faulty sequence. Because we have seen that whenever we go for any gene therapy, so this CRISPR-Cas9, ZFN, TALEN, they make double strand breaks. And after that, the DNA repair takes place in which different outcomes can be achieved depending on that which repair mechanism is active. So in this case, homology dependent repair gets active if we have the homologous DNA which is present. So in this case, we artificially provide a donor DNA sequence. So that's why which can act as a template to correct the faulty sequence. So recombination takes place at that site and we repair the faulty gene. And whereas option A was combine CRISPR RNA to guide Cas9. So this is not true because Cas9 is the one which is guided by the guide RNA and not the donor DNA. And then option C was create a double strand break. So double strand break is caused by the Cas9 gene and not by our donor DNA. Then activate the PAM site for Cas9 recognition. So this is also incorrect because PAM recognition depends on the target DNA sequence. So these options are just to confuse you with the target DNA or the guide DNA. So always keep this thing in mind that the donor, whenever donor word is written, so donor word comes when we are doing the homology or homologous based repair. Okay, so I hope you won't get confused in future. So next question is, which skid types are exceptions to typical autosomal recessive or X-linked inheritance patterns? Option A, Digeot syndrome and certain cases of Hoyral-Heredderson syndrome. So sorry. Option B is all autosomal and X-linked forms of skid. Option C, only the Digeot syndrome. Option D, only the Hoyral-Heredderson syndrome. Okay, so need to tell which one is the exceptions to these. Because when we were talking about skid, then we have seen that the inheritance pattern is either autosomal recessive or either it's X-linked. So we need to tell which one is the exceptions. Okay, now let's see. So the right answer is option A, which is Digeot syndrome and certain cases of Hurrell-Hradson syndrome. This is correct. These are the exceptions as they are not caused by single gene mutations but involve developmental defects and complex inheritance. So these are exceptions. Next option was all autosomal and X-linked forms of SCID. So it is completely incorrect because they are mentioning themselves that all autosomal and X-linked forms. Next option was only the Digeot syndrome. So this was incorrect because this Hurrell-Hradson syndrome also shows this particular inheritance pattern. And similarly, in last option also, they have forgotten the Digeot. So that's why these options are not correct. And option A is correct because both these forms are complex forms which shows atypical inheritance. Next question is mutation in which gene leads to T negative B positive SCID, where T and NK cells are absent but B cells are present. So it's clear from this name only. T minus B positive, NK minus SCID. Option A RAG1 and RAG2. Option B IL2RG. Option C ADA and option D is PRKDC. So you need to tell mutation in which gene leads to this. So just think in the term that it is affecting just the T cells and NK cells, but not the B cells. So if you know which gene is responsible for the B cell defect, then you can cancel that out and then you will be able to get the answer. Okay, now let's see. So the right answer is option B, which is IL2RG. If we talk about RAG1 and RAG2, so this also leads to defect in the B cells. So T negative, B negative SCID. In case of IL2RG, mutation in this gene, common gamma chain, it leads to X-linked SCID where the T and NK cells are absent but B cells are present. If we talk about ADA, this is most severe form of SCID in which all of them are absent. If we talk about PRKDC, this is incorrect because mutation here also leads to T negative, B negative SCID and not the T negative, B positive. So now keep this thing also in your mind that when we were talking about the question that which is the most severe form of SCID, so why it is most severe ADA? Because in this all these cells are negative, all these cells are absent. That's why this was the most severe form. And if we talk about the form in which T negative and NK negative is there but B is positive, that is IL2RG. So now let's move to next question. Next question is which of the following correctly differentiate hemophilia C from hemophilia A and B? So option A is hemophilia C is autosomal, whereas A and B are X-linked. Option B, hemophilia C is due to the factor 8 deficiency, while A and B are due to deficiency of factor 9. Option C, hemophilia C is more common than hemophilia A, but less common than hemophilia B. Option D is hemophilia C only affects females, while hemophilia A and B affects only the males. I think this will be the most easiest question in today's session because we have discussed it a lot. A and B and C forms of hemophilia. Okay, now let's see what is the right answer. So the right answer is option A. So in which the hemophilia C is the autosomal, whereas A and B are the X-linked. So this is correct. And in this, they mentioned that hemophilia C is due to factor 8. So this is also wrong because A is due to factor 8 deficiency, B is due to factor 9 deficiency, and C is due to factor 11 deficiency. So this option was wrong. Hemophilia C is more common. So this is also wrong because hemophilia C is the one which is most rarest form, whereas hemophilia A is the most common, and then hemophilia B. Then it says it only affects females. So till now I have not seen any such defect which is only affecting the females, other than if it is X-linked dominant inheritance or if it is the dominant autosomal inheritance if it shows, then only it can affect the females. Otherwise, how will it affect the just the females? While A and B only affects the males. So this is just to confuse you so that you think that in the X-linked recessive, mostly males get affected. So maybe this could be right, but no, this is not right because this is completely wrong statement because hemophilia C can affect both the males and females because it is autosomal. So it is not biased towards females. So keep this thing in mind. Now let's move to next question. So next question is which inheritance pattern is most commonly observed in SCID? Option A autosomal dominant, option B autosomal recessive and X-linked recessive, option C mitochondrial inheritance, and option D is Y-linked inheritance. Okay, so this is also the most easiest question and this is quite straightforward. I don't think we need explanation for this. Okay, so the right answer is option B and it is self-explanatory. We have already discussed it. Okay, so this is all about today's session. So I hope now you might have cleared all your doubts related to the week 8. And if you still have doubts, then you can ask me. So let me see. So we still have some time. So I have some more questions. So we will discuss that also. And these questions which I have kept here, so these are from the previous year assignments, and these might come in your exam. So if you're not attending the live class, then if you're watching this video, then also practice these MCQs because even if you skip the topics, but if you go through these MCQs, then it will help you to refresh the topics. So it will make you revise all those topics. Instead of reading them again, you can simply go to MCQs and you'll clear all your doubts at once. Okay, so now next question is, what happens in ADA, which is adenosine deaminase deficiency, related to SCID? Option A, lymphocytes multiply uncontrollably. Option B, toxic metabolites built up, causing lymphocyte death. Option C, cytokine signaling improves, boosting the immune response. Option D, thymus develops normally but B cell function decreases. So you need to tell this ADA deficiency is related to. What happens here? Okay, now let's see. So the right answer is option B. It leads to toxic metabolite buildup, causing the lymphocyte death. Because we have seen that how the adenosine monophosphate get accumulated in the deoxyadenosine get accumulated in the toxic amount, and other toxic metabolites also increase, which are toxic to lymphocytes, which leads to the imbalance in the lymphocyte population and kills them. So this was the right answer. Whereas if this is in this, they are saying cytokine signaling improves, boosting the immune response. This is completely wrong because it is impaired in the case of ADA. Because we have seen that ADA is most severe form in which T negative, B negative, and NK negative forms are observed. So T negative means cytokine signaling is affected. Thymus develops normally. This is also wrong because in this thymus, T and B, both the cells are affected. And this is also incorrect because lymphocytes do not multiply, they do not proliferate, they get depleted. There is death of lymphocytes. Okay, now let's move to next question. So next question is hemophilia A occurs due to the deficiency of which clotting factor? So option A is factor 9, option B is factor 8, option C is factor 11, option D is factor 5. Oh, okay, so till now we have discussed it many times. So I hope now you know which will be the right answer. Okay, let's see. So right answer is option B, that is factor 8. Factor 8 is the one which leads to hemophilia A, and this is the hemophilia A is the type of hemophilia which is most common. Factor 9 leads to hemophilia B. Factor 11 defect leads to hemophilia C. Factor 5 is incorrect because this deficiency leads to rare bleeding disorder which is known as parahemophilia and not hemophilia A. So keep this thing also in mind because from this slide you learned this new thing which is parahemophilia. So factor 5 defect leads to parahemophilia. Now let's move to next question. Next question is, which clotting factor is missing or reduced in hemophilia B, also known as Christmas disease? Option A factor 8, option B factor 9, option C factor 11, and option D is factor 7. So this also we have discussed just now. So right answer is option B, that is factor 9. Next question is, severe hemophilia is diagnosed when factor levels are. So in this, it is severe hemophilia. So option is more than 50 IU per DL. Option B, between 5 to 50 international unit per DL. Option C, between 1 to 5 IU per DL. And option D is less than 1 IU per DL. So this is also straightforward. I'll give you a little hint that they are saying severe hemophilia. And when the factor levels, so in this they are talking about that how much amount of the factors are present in the blood or in the cells. So which means that you need to tell that the factor 8, factor 9, factor 11, which leads to the different types of hemophilia. So out of them, the severe hemophilia, so in which, what is the percentage, what is the amount of those factors in the blood? Okay, now let's see. So the right answer is option D, which is less than 1 international unit per DL. Because if the amount is very, very low, then only the blood will not get coagulated and it will lead to hemophilia. So keep this thing in mind that in the case of severe hemophilia, the concentration, or you can say the activity, is less than 1 international unit per DL, which leads to frequent spontaneous bleeding. Whereas 1 to 5 is the moderate hemophilia, 5 to 50 is the mild hemophilia, and 50 international unit per DL is the normal or mild form. Okay, so with this, let's move to next question. So next question is, the primary role of factor 8 in blood clotting is to. Option A, act as a cofactor for factor 9A to activate factor 10. Option B, break down fibrin clots. Option C, directly convert prothrombin to thrombin. And option D, activate the platelets for wound healing. So you need to tell the primary role of factor 8. Okay, now let's see. So right answer is option A. It acts as a cofactor for factor 9A to activate factor 10. So this factor 8, it forms the complex with factor 9A on phospholipid surface to efficiently convert factor 9 to factor 9A in the coagulation cascade. So when we were talking in the session, then we looked into a cascade, that how these different factors they initiate the series of reactions. So they activate each other, and ultimately factor 1 is activated, which generates the fibrin clot. So with that, it is a series of reactions. So there is a cascade going on. Option B was break down fibrin clots. So fibrin clots are not broken down, they are formed. And this breakdown is mediated by the plasmid and not the factor 8. Next was directly convert prothrombin to thrombin. So this is incorrect because factor 9 is activated to factor 9A in combination with factor 5, and then they does this and not the factor 8 directly. So factor 9A and factor 5 leads to this direct conversion of prothrombin to thrombin. Last option was activate platelets for wound healing. So this is incorrect because platelet activation is independent of factor 8. Factor 8 functions in just the coagulation cascades. So it does not activate the platelets. Okay, let's move to next question. So next question is, what is the main approach in genome editing therapy for SCID? Option A, fixing faulty immune genes in the patient's stem cells outside the body and then reinfusing them. Option B, introducing new genes into the bacteria to produce missing proteins. Option C, using drugs to increase T cell numbers. And option D is replacing the entire thymus with a donor thymus. So you need to tell what is the main approach in genome editing therapy. So it should be clear from the name genome editing. Okay, so now let's see. So right answer is option A, which is fixing the faulty immune genes in patient stem cells outside the body and then reinfusing them. So this type of approach is known as ex vivo gene editing, where we have seen that how the hematopoietic stem cells were obtained from the patient, they were treated, and then the corrected copies were inserted so that they can give rise to the cells which were defected previously, such as B cells, T cells, natural killer cells. Now, second option was that introducing new genes into the bacteria to produce the missing proteins. So, what we will do by producing missing proteins in bacteria? So, this option is not right because this is the protein replacement therapy and this is not the genome editing. So, in this case, for example, in case of hemophilia, when we have seen that we supply certain genes which give rise to the um, duplicated, which mimic the factor 8 activity. So in that case, we do the protein replacement therapies. So in this case, it is not right. Then option C was that using the drugs to increase the T cell numbers. So this is also incorrect because drugs cannot fix the underlying genetic defects. And this does not come under genome editing because this will not edit anything in your genome. And then next was that you replace the entire thymus with the donor thymus. So this is completely incorrect because this is known as thymus transplantation. It is experimental and it is not the genome editing approach. In this, you're not doing anything with the genome. So this option you cannot even think of. This is completely wrong. So let's move to next question. Next question is genome editing therapy for hemophilia aims to option A, modify platelet producing genes. Option B, correct or replace the genes for factor 8 and factor 9 production. Option C, target red blood cell production genes. Option D, inhibit thrombin activity. So you need to tell that the therapy which we are using for hemophilia, what it will do, what it aims to. So this is straightforward only because if you know that how the hemophilia happens and what if treated then it can cure hemophilia, then you will be able to answer this. Okay, so let's see. So the right answer is option B. That is correct or replace the genes for factor 8 and factor 9 production because we know that factor 8 leads to hemophilia A and factor 9 leads to hemophilia B. So if we can correct them, then we can treat hemophilia. And this comes under the genome editing therapy because we are editing these genes. Option A was modifying the platelet producing genes. So we have previously also discussed that the platelets are not the primary targets in hemophilia therapy. And then C option was that target the red blood cell production genes. So this is also incorrect because hemophilia is a clotting factor disorder and it has nothing to do with the RBCs. Then inhibit thrombin activity. So this is incorrect because thrombin should not be inhibited. It should be activated. So prothrombin is converted to thrombin, which ultimately leads to the clotting. So if we will inhibit the thrombin, then it will worsen the bleeding and it will not treat the hemophilia. So that's why right answer is option B. So we have next question, which is which gene defect is most common in X-linked SCID? Option A, ADA, option B, JAK3, option C, IL2RG, and option D is RAG2. So keep this thing in mind that it is X-linked SCID and it is not autosomal. Okay, so let's see. So right answer is option C, which is IL2RG. Now, if we talk about ADA, although this is the severe form of SCID, but it is autosomal recessive SCID and it is not X-linked. If we talk about JAK3, this is also incorrect because this deficiency also causes autosomal recessive skid. That means this gene is present on the autosome and not on X or Y chromosome. If we talk about IL2RG, so this is correct because this gene is present on the X chromosome. That's why it is most frequent cause of X-linked skid. If we talk about RAG2, this is also incorrect because these mutation causes T negative, B negative skid, which are usually autosomal recessive. So let's move to next question. Question number 29 is, why is iron chelation therapy necessary for thalassemia patients receiving frequent transfusions? So option A, to prevent extra hemoglobin formation. Option B, to reduce excess iron buildup caused by repeated transfusions. Option C, to boost production of alpha globin chains. Option D, to stabilize red blood cell membranes. So if you remember, we have already discussed one such question in which we discussed that what will happen if the person is receiving the um, long-term transfusions. Okay, now let's see. So right answer is option B. That is to reduce the excess iron buildup caused by the repeated transfusions. So in option A, it was saying that to prevent the extra hemoglobin formation. So this is incorrect because we are not getting the iron chelation for this reason. Transfusions do not cause the extra excess hemoglobin production. This second option is right because there is excessive iron buildup due to repeated transfusions. So we need the iron chelation to avoid that iron overload and prevent the organ damage. Option C was to boost the production of alpha globin chains. So this is also incorrect because chelation therapy does not affect the globin chain synthesis. Option D was to stabilize the red blood cell membranes. So this is also incorrect because chelation does not directly stabilize the red blood cell membranes. So right answer is option B. Now the last question for today's session is, the CRISPR-Cas9 tool is primarily used in gene therapy too. Option A, cut DNA at precise locations to allow correction. Option B, directly repair damaged blood vessels. Option C, create random mutations to study gene function. And option D, remove the defective chromosomes completely. So this is not this is not difficult at all because till now we have discussed a lot about genome editing methods, genome editing tools. So I hope you know the answer. Okay, let's see. So right answer is option A. Cut DNA at precise locations to allow the correction. So it introduces the double strand breaks at specific DNA sites, which enables the correction by the repair mechanisms like HDR. So if it is undergoing HDR, then we supply the donor DNA, which will carry the correct copy of the gene. Or for example, if we want to mutate some gene, or if the gene is already mutated, then it depends on that. We will supply the respective donor DNA and it will undergo the precise correction of that particular gene. Second option was that directly repair the damaged blood vessels. So this is incorrect because CRISPR acts at the genetic level and not on the tissues like blood vessel directly. Option C was like create random mutations to study the gene functions. So this is not a random mutation because we are not studying gene functions using gene therapy. From the name only, it's clear. Therapy means we are doing something to treat something. Therapy means which is treating something, which is correcting something. So it is a precise correction. It is not random. Option D was remove defective chromosomes completely. So this is incorrect because we are not removing or doing something at the larger level. So this modifies specific gene sequences but not removing the entire chromosome. We can change some genes, but we cannot delete the entire chromosome or remove it completely. So the right answer is option A. Okay, so this is all about today's session, and I hope the concepts are now clear. So if you still have some doubts, then you can write it in the comment section or you can put it in the discussion forums. So from there, I'll be answering your doubts. So in the next session, we look into the week 9 topics. Till now, I'll end the class. So thank you very much for listening and have a good day. Bye.